biology. So we've finally made it down to our last bit about membranes. So in this section, I want to focus on the movement of larger substances or debris across the membrane. Now I have two words up here. I have exocytosis and endocytosis. So as we've done before, let's use the word to define the word. If I have CYTE, I'm referring to a cell, and then exo is to exit. So literally as a word, we're talking about exiting the cell. So let's talk about how we would do this, because we're talking about a secretion of a large molecule. It is going to do this by fusing or having a fusion of a vesicle, and that's going to actually happen with the plasma membrane. So I'm actually going to start with the cell and drawing the cell out, which we'll get to and talking about as well. But if I draw the nucleus has a membrane, and then we have a continuation of this membrane, which I'm drawing the endoplasmic reticulum. Now here we're making proteins and we have the smooth endoplasmic reticulum, and we'll focus on that when we get to the cell, but it's making stuff, okay? It's going to go to the Golgi apparatus, where that Golgi apparatus is going to package and get ready for shipment, and kind of like the UPS or now my Amazon. And this is going to fuse off with a vesicle. And this is all made of membrane, because we're going to have this idea of this continuous membrane that we're going to talk about with the cell. This vesicle will eventually, and I'm going to leave a space over here because I need some endocytosis. So this really is a full circle and will end up being a full circle. But this vesicle will end up fusing with that plasma membrane to create kind of a pocket in which that material can be excreted from the cell. So that's our exocytosis. So things are moving out of the cell. Now, in an endocytosis, okay, it takes macromolecules and debris by forming vesicles into the plasma membrane. So let's say that there's some debris out here. Okay, we will actually see the plasma membrane somewhat wrap around this stuff. This will eventually pinch into the cell, where we bring those particles into the cell. So now we have an endocytosis. So endo meaning within and cyte meaning cell. So within the cell. So if we want to just even simplify in these terms, endocytosis, we're bringing stuff in, and then exocytosis, we're bringing stuff out. All right, now I have two specific types of endocytosis. We have a phagocytosis. There's that CYTE again. And then phage is to eat. So we have literally eating from the cell. And what we're talking about is large amounts of like solid stuff would be taken in to make that vacuole. So phagocytosis would be endocytosis with solid stuff. Now, penocytosis, we're talking about taking stuff in, but what we're taking in is going to be water. So again, it just has a specific name or a special name, like osmosis in our last set of notes or our previous set of notes. Okay, so a large amount of fluid is actually taken in. Okay, now I like this final picture, and this is great closure on our plasma membrane of what we've been talking about. So we see this phospholipid bilayer. Here's all of these proteins. We see that transmembrane protein or integral protein. Here's that peripheral protein that I told you guys I'd have a picture of for you. We have those channel proteins that we see here. The last two things that I want to mention are going to be carbohydrates or oligosaccharides that are actually attached or bonded to some type of protein or lipid within the plasma membrane. And the first one is going to be a glycoprotein. Okay, so we see that glycoprotein that's right here. And we kind of see this as almost looks like an antenna as it's sticking out of the cell. The function of that glycoprotein is to serve as a receptor and enzymes. Okay, so it's actually communicating. So when I said antenna, it kind of literally is in terms of what it's communicating with the cell. Then we have the glycolipids. Okay, so the first one we had a glycoprotein, now we have a glycolipid. And these are going to serve as markers. So an example where we would see that would be like the blood groups on red blood cells, so the ABO blood groups that we see there. So this is our final set of notes going over the plasma membrane that I have so far. Be sure to look for a set of notes. I'm going to do a specific set of notes for the lab this time in explaining that movement of the concentration of where we see that hypertonic and hypotonic and isotonic. But I want to put that with the lab portion because that fits a little better than actually putting it within the notes section. So look for that within the lab. So your layout's going to look just a little different because I'm going to embed that before your lab. Okay, and as always, let me know if you have any questions.